Hello and welcome grade 10 life science mindsetters to today's life science exam school show. I hope you guys have been studying hard for these upcoming end of year exams and I hope you guys are keeping well. Today in studio we've got Cheryl. How are you doing, Cheryl?
Fine, and you, Lloyd?
I'm all good. So guys, before we start, remember this show is sponsored by the MTN SA Foundation. Thank you very much to them, as well as in being endorsed by the Department of Basic Education. So guys, before we start, all the notes from today's show can be found on our Facebook page. That's facebook.com/learnextra. So all those notes, as well as the challenge question, which we'll be going through now. So guys, before
We're going to start. We've got so much that we have to do.
We have a lot to do in this lesson, so let's do it.
Okay, thanks, Lloyd. Welcome, grade 10s. Oh, the crunch time is here. We've been talking about it the whole year. I'm afraid next couple of weeks, no TV, all right? I'm writing grade 4 exams at the moment, and I'm stressing, so I don't know what you guys are going to be going through. Okay, we're starting with Life Science Paper 1, all right? And most important thing that you need, all right, and I've put it, I'm going to show you quickly. Those of you, if you can go and get the notes, please. The most important thing when you write your papers, to know what is going to be in paper 1 and what is going to be in paper 2. This week we're looking at paper 1, so you need to have the following: biochemistry. All right, and I think when I've done the questions, I've actually put that in order. So 1, biochemistry, which is your proteins, carbohydrates, fats, your enzymes, your micronutrients, your macronutrients, your vitamins, all right? Your food test, very important. Then you need to know the cell, all right? The structure of the cell, mitosis, osmosis, cancer, cloning. All of those are part of the cell. Then we looked at tissue. You need to know plant tissue, which is your sclerenchyma, your parenchyma, your xylem, your phloem. Your mammalian tissue, all right, epithelium, nervous tissue, muscle tissue, cartilage, ligaments, bone, blood. All right, and then you need to know your plant organs. So we need to know the root, the stem, and most importantly, the leaf. Okay, so when you start your studying, make sure you know what is in paper 1 and what is in paper 2. Okay, so now we're going to start. I want to get into those questions. The only way that you are going to be is exam ready, 1, if you've studied, all right, that is duh. And 2, you need to do past papers. Find out from your teacher. I don't know if you've got access to libraries at school. Go onto the Mindset page. See if you can find any kind of past exam papers. Go over, get from your friends, whatever. Try and practice as many papers as you can. All right, so we're going to start now. All right, guys, the first question is a bit of a challenging question, all right, but I want you, we're going to start off with biochemistry. Biochemistry, one of the things that I think is the most important is enzymes, because the enzyme section relates to almost everything that you are going to do. Okay, so here, what I would, for each of your sections, what I would suggest, how you study might be up to you. Have a nice big piece of paper, an A3, all right, and you have there carbohydrates, proteins, fats. All right, and then tell me things like, what is the structure for each of them? What are the food tests for each of them? What happens during the food tests? What are their bonds? Each one have got different kinds of bonds. All right, what are their elements? What do they build? Right, in other words, what are they going to ultimately, why do we eat all these things? Okay, so we're going to ask you 2 questions here. The first one is an enzyme question. It is a bit challenging, and then I'm just going on to a basic cell question, and we'll go through it to see what time permits. Okay, this question 1, let me get our pen going. All right, come pen, oh, there we go, and we know I like pink. Okay, an investigation was conducted to investigate the effect of different temperatures. All right, and pH levels on the activity of an enzyme. All right, now you need to, warning bells going off. Temperature and pH of enzymes. What words do we need to start thinking about? There's 3 words. All right, what enzymes are sensitive to temperature? Enzymes are sensitive to temperature. Enzymes are sensitive to pH. Because what is an enzyme? An enzyme is a protein. All right, and when it comes to enzymes, there's 3 words: inactive, optimum, which is best, and denature. All right, so we're doing an experiment where we've got, all right, a specific enzyme, and what we're going to do, it's going to be exposed to different pHs and different temperatures. And we then need from that, we're going to be able, all right, to read from our graph. All right, now comes the catch, all right? In each case, the enzyme was provided with 12 grams of substrate. Substrate is food. In other words, what it acts on. All right, what it acts on. Okay, so we've got 12 grams of substrate. To break down to the complete breakdown, so if we have, all right, the substrate which is whole, 12 grams, all right, enzymes are biological catalysts. They break down things. Our products, our end product, our products must also be 12 grams of product. Okay, so if I started off with 12, I need to finish off with 12, but they need to be changed. I need to break them down into smaller parts. Okay, now here we go. Let's have a look at this graph. On first sight, it looks a bit daunting. All right, but let's have a look what it says. Okay, the graph shows the effect of temperature. Okay, so here we've got 3 graphs. We've got 3 line graphs, 1, 2, 3. Okay, here is the range of temperature. What do I need to look at? Those words again: inactive, optimum, and denature. Okay, then the 3, they were exposed to different pHs. Here, number 4, all right, they are slightly acidic. This one over here, all right, is slightly, we can almost say it's neutral. Right, and this pH over here, right, let's get to it with the blue, that one over there, that is my alkaline. All right, what do we know? All right, enzymes are pH specific and substrate specific. Now my first question. With reference to the graph, it means read what I can see from the graph. All right? Read what I can see from the graph. You don't make up your own general knowledge. Lots of you think, oh, the first thing I must do, I must go for body temperature. Body temperature is the optimum. No. All right, let's have a look here. It says, all right, state the optimum temperature and pH of this enzyme. We are looking for which of the enzymes, all right, works best. What does optimum mean? Best. All right, temperature and pH of this enzyme. So let's go. Let's have a look at temperature. I'm looking for peaks. All right, I'm looking for peaks. Remember, that's what I'm looking for. That is my optimum. That is my inactive. When it doesn't work anymore, that is my denature, changes shape. So what do? I take my graph and I see here that this enzyme, all right, has its peak. So which pH is pH number 4? And if I draw a line downwards, right, with a ruler, obviously, all right, I'm going to see that it works best at 36 degrees. So with reference to the graph, state the optimum temperature, 36 degrees Celsius. There, please don't forget the units of measurement. And a pH, don't tell me just 4. 4 means nothing. You could have 4 cats, 4 dogs, 4, all right, toes on 1 foot. A pH of 4. And that's where you get your 4 marks from. Okay, so where does it work best? That's what I'm looking for, where I have the peak. And this one works the best out of all of them. Okay, now there's a bit of a long question here. At each temperature and pH level, the enzyme was allowed to react for 20 minutes, okay, before the amount of product was measured. Was the enzyme ever working effectively enough to break down all? Okay, let's go back to the beginning question. So it says here, was the enzyme ever working effectively enough? So what do I want here? I'm going to answer a yes or a no. Then for my other 2 marks, do not forget to carry on reading. Explain. Okay. Remember I said to you, let's go back. This is where the maths comes in. You started off with 12 grams. If it was completely broken down, the product also had to be 12 grams. Have a look here. Grams of product. This is what we're looking for. Did any of them reach this point? Did any of them reach them? It says it broke down all. To break down all, one of them must have been 12 grams of product. Did we reach it? No. All right? No. There's your 1 mark. Remember I said, working, you must give me a yes or no first, then we explain. All right? And your answer is no. Why? Okay, there, explain your answer. That means why? Because no enzyme broke down the product to 12 grams. The pH 4 was close. That was 10, but the question was all. Broke down all. All right? And there we saw there was none of them. The product wasn't at 12 grams for any of the enzymes. So obviously, no, it wasn't a very successful, all right, enzyme. Explain why the enzyme showed no activity at 45 degrees. Now, if we go back to our graph, right here, 45 degrees, why does it not show any? What is the word? Let's go back to our graph. What happens at very high temperatures? Okay, explain. Explain. So 45 degrees is a high temperature. All right, what happens at high temperatures? My enzyme denatures. Okay, and remember, denatures means it changes shape. And because it denatures, it can't. All right. Remember that lock and key? It can't lock with the substrate. And if it can't lock for the substrate, therefore it can't break it down. Remember enzyme, lock and key. All right. You need to have that. Now, very important. In this investigation, name the following: the independent variables. That was your key. All right. Independent starts with an I. What did I choose to use? And I chose the pH, and I controlled the temperature. All right. Dependent, all right, is your results. Now, if you go to your graph also tells you, what do I find on my x-axis? Independent. What do I find on my y? Dependent. What is my dependent variables? Grams of product. All right? Grams of product. Grams of product. Okay, also read your questions. Variables, 2 marks. Okay. All right, over here, 2 controlled variables, i.e. the variables that was kept constant. All right, what did you have to keep constant? Don't tell me pH and temperature. Don't, because those are the things that you changed. All right, what must you make sure that was the same all of the time? Okay, and let's go, when it comes to practicals, you need to think of this. What did I need to keep all the time? 1, I needed to keep my substrate. What did I act on? I couldn't put one with potato and the other one with margarine. Doesn't work. All right, so I had to have the same substrate. What else did I have? The same enzyme. All right, you couldn't choose. You said an enzyme. All right, so we need to have, all right, the same enzyme. Last question on this point, all right, from the graph. From the graph, so they're asking you, go to the graph again, determine the amount of product produced at a temperature of 20 and a pH of 4. You go to your graph, you use your ruler, okay, and what do? Here's my pH, here's my temperature. I draw a line, straight line, up till I reach there, my pH. Then with my ruler, I draw another straight line because it wants me to give grams, all right? And there we go, that is my answer. And I notice it's almost close to 4, so I can give an estimate, all right, of approximately 3,8 grams. Are we that for the first segment?
Yeah.
Time flies.
Yeah,
so
guys,
quickly.
time is going quick. I hope you guys are enjoying this lesson so far. I hope you guys are keeping with it. If you're not, if you have too many questions and you just can't get to the bottom of it, remember you can always go to our help desk. But guys, let's take a quick ad break and we'll see you right after the break. Hello and welcome back to Life Science Grade 10s. Hope you guys didn't miss the first segment, but if you have, there's still catching up to do. You can go to the facebook.com/learn extra page. All the documents and papers that we are working on right now can be found on that page. But without further ado, let's carry on.
We're rushing. We've got so much to go through. All right. Some of the questions I'm not going to answer all of them. I'm just showing you the different kinds of questions that are typical, all right, for that section of work. The next question on biochemistry, all right, be prepared to answer. Quite often they like to use some type of foods label of what kind of food is in, or what a breakfast plan or a meal plan or something. Remember your carbohydrates, your proteins, your fats, all your vitamins and all your nutrients. You can bring that into the question. So I've got like a little one over here, all right? I just want to show you the kind of things that we can bring into it. And the question is, the following table shows an analysis of nutrients. Oopsie, there we go. Found in a 100-gram portion of breakfast. The breakfast was made up of eggs, bread, and butter. All right. Now, ready? Eggs. We need to start looking. Protein. If you did your food tests. Bread. We're looking at carbohydrates. All right. So that's your starch and your glucose. And your butter, we are looking at your fats. All right. And if we have a look, all right, at the other nutrients, there we've got, those of you, if you didn't realize that, carbohydrates, fats, and proteins. Then we've got our nutrients. We've got calcium and we've got iron. Calcium for building bones, iron for our red blood cells. All right, we've got vitamin A for our eyes, and vitamin C, all right, for helping us build healthy our teeth and our blood vessels, etc. Okay, so already here, I should be able to tell you again what kind of questions are going to be. Once again, it's a data response. I actually give you the information, and you now need to give me the appropriate response. Okay, it says here, use the information in the table. Please don't go and try and use your own initiative. It asks you, what does the table tell us? Okay, now question number 6. Name a vitamin deficiency. All right, now that should narrow it down because you've got 2 vitamins there. You've only got 2, vitamin A and vitamin C. That could result from a diet that consists mainly, all right, of eggs. Now, vitamin deficiency caused by disease mainly of eggs. Now, if we have a look here, look, a deficiency. eggs are rich in vitamin A. So do we have a deficiency? No. But what do eggs not have? So if I only ate eggs, I wouldn't get any vitamin C. All right? And if I don't have vitamin C, what is a deficiency disease? Yes, I'm sure your teachers showed you all scary pictures. Scurvy. All right, your teeth start to fall out, the blood vessels. All right, not very pretty at all. Now, question number 7. Identify the food type. So what they want for you here, the food type, they're not asking for the vitamin. You had 3 food types. Your answer is egg, bread, or butter. One of the 3, okay? Now we carry on reading. That would be best for a person who suffers from night blindness. Now let's go back. All right, it says identify the type that would be best. Best. That is what you need. All right, so which food, all right, has a person who suffers from night blindness? Now, night blindness has got to do with the eye. And when it comes to the eye, we need a vitamin rich in vitamin A. So let's go and see. We want to look at the food groups. Let's have a look. There's vitamin A. All right, my choice is egg, 675. Okay, fair. Bread, 12. Oh, bread's out. Okay. Wow, butter, 2,344. Okay, so what is my choice? If I were to suffer from night blindness, right, I need to get more vitamin A in, and what am I going to read there? I'm going to eat more butter. Okay, and it says, give a reason for your answer. Because it is rich in vitamin A. And what is vitamin A? Vitamin A helps with our eyes, especially the retina. Okay, the cones in our eyes. And rods, should I say more the rods. Which food type, again, which food type, what are you looking for? Bread, butter, or eggs will show, now this is where your food tests come in. No color change when it is tested with biuret solution. So in other words, biuret, what do we use there? Protein. Protein. My biuret was my protein test. So which of the food has no proteins in? None whatsoever. Right, let's have a look. There's protein. Okay, so if I test my eggs, I'm going to get, it has protein. What color is it going to go? Yes, that violet purple. Bread has a little bit, going to go slight, 0. Butter might be rich in vitamin A here, but it definitely, all right, does not have any proteins. It says no color change. Okay, if we were doing the color test, it would leave a nice little spot. This breakfast provides very little vitamin C. What food would you recommend to correct this? It's a breakfast, okay? So any kind of what is rich in vitamin C, here you can, your imagination, all right? What do we know? Oranges, all our citrus fruits. Oranges, kiwi, all right, strawberries, pineapple, all right, only one. So we know what food is rich. Spinach, all right, gross, but anyway, all right, what food is rich? There we go, spinach. Okay, any of these for 1 mark. Okay, the next question should be familiar. I'm not going to go too much into, all right, this question. It's a basic diagram. You should have the cell. The cell, by now, you should know off by heart, all right? So when it comes to questions with the cell, all right, the first thing that you need to recognize, is it a plant or an animal cell? Having a look over here, you will see it is an animal cell, and your reasons are it only has a cell. Oh, here we go. That only has a cell membrane. All right. And what else do I see? I don't see any chloroplasts. so I know it can't be a plant cell. All right? So always know, is it a plant? Is it an animal? Give reasons for your answer. Be able to label the organelle. So if you're just looking here, you need to be able to say, okay, this is the mitochondria. All right, that is the nucleus. Okay, that is, it's got probably endoplasmic reticulum, and it's got ribosomes on it, so it's rough endoplasmic reticulum. This is the Golgi apparatus. Different, always looks like a stack of pancakes. These are probably ribosomes. All right, okay, so be able to label it and to know the functions. Even here, not labeled, centrioles. All right, so when it comes to the cell, be able to label the cell. So you have a piece of paper, I call it annotated diagram. You've drawn a nice picture of the cell, and you've got labels all over the show, and next to the labels, you've got each of the functions. Mitochondria produces energy. Chloroplast for photosynthesis. Vacuole stores food. Nucleus controls all the activities of the cell. Okay, now I want to go into a bit more of a difficult question on this section. All right, and that is the mitosis question. You might get a variety of diagrams, and from the diagrams, you need to be able to differentiate which phase is which. Okay, now I always remember, all right, IP-MAT. And what does IP-MAT stand for? Interphase, then prophase, then metaphase, then anaphase, and then telophase. All right. So if we have a look at our diagrams over here, we have got 4 cells. Let's see if we can now put them into place. Okay. First thing is interphase. Interphase, I can't see any kind of chromosomes. It looks like the cell. So here I can see chromosomes quite visibly, so I know there's no interphase. Prophase, all right, what starts to happen in prophase? The cells start, the chromatin network, all right, loses its water, it thickens, and I start to see chromosomes. My cell membrane, all right, is still there. My nuclear membrane is still there. So let's have a look. All right, I see here, look here. Right, what do I start to see? I can start to see. So I know that this is prophase. Okay, then the next phase is metaphase. For middle. Do I see any of my chromosomes that line up in the middle? There we go. Nice and clear on the middle of the equator. Anaphase. Anaphase, they get torn to the poles. What do I see here? All right, each set of chromosomes going to the poles. And my last phase is telophase. Telophase is when my division is finished, cytokinesis occurs, and what have I got? 2 cells forming. All right, so we can call this telophase, or if you want, after division, cytokinesis. Okay, so now we have, all right, we've labeled the different phases. Let's see if we can answer the questions. First question relates back to cells. How do you know that these are plant cells undergoing mitosis? Give 2 reasons by referring to the graph, the pictures. You can't have a look here. What do you see? All right. The first thing that you should notice is this. What do we know about a plant cell? The 2 things. I can think of maybe 3. The 2 things. Why do we know it's a plant? 1, it's a regular shape. And number 2, did you notice something else? It had a cell wall. All right. The third reason, and it's not always visible, is that plant cells do not always have spindle. But have a look here. All right, what can you see over there? It's a spindle fiber, so we're not going to use that, all right? So don't use that as your question. Sometimes they don't have, all right, and sometimes they do. Spindle is very clear here. So what do we see? All right, we can see that it's a plant cell because plant cells usually have a regular shape, and what do we see? we can see that there is a cell wall. Okay, next one, label structure 1. It's a chromosome. Okay, not a chromatid anymore, a chromosome. Number 3, explain the role. So tell me why of structures labeled 2. All right, what is number 2? They are the spindle fibers, right? And what is their function? All right, remember when we talk about the spindle fibers, I always say it's like a spider. Spindle fiber, spider, all right? And what does a spider spin? It spins like a web, right? And when it spins like a web, it gets stuck in between the two, all right? And what does that spindle do? It actually pulls, right? So if one side was to pull the spindle one side and the other, what does it help us do? It pulls our chromatids apart. All right, so let's try and be more biological, explain the role of the structures, label them first. I know it's only for 1 mark. They are the spindle fibers. And what is their function? They pull the chromosomes towards the poles. All right, so remember, they're the ones, I also imagine they're like fishing rod. Spindle fiber, imagine the gut for fishing rod. Right, and you're just going to pull. Okay, if you have a look here, it says, name the phases of cell division that cells and C are each undergoing and give a reason in each case. Okay, so now it says, name the phases. We've done that. Right, so you put A and you there, and you put C there. And they want 2 things for you. 1, name them, and 2, how, all right, were you able to identify each phase? So how did you know that this was metaphase? I knew it was metaphase because where do the chromosomes? They lie at the equator, all right? Remember, M stands for middle. They're in the middle of the cell. Number C, why did I know it was prophase? I know it's prophase because what can I still see? I can still see the nuclear membrane. And what do I notice? The chromosomes are just separating. All right, so when you get to this, you need to be able, are we time for a break, Ogedi?
Yeah.
I think the 15 minutes
Flies by.
Flies past.
So guys, before we carry on, let's take a quick break. Stretch your feet, stretch your legs, take a little glass of water, but we'll be back in a few minutes. Welcome back, grade 10 life science pupils and mindsetters. I hope you guys haven't missed the first 2 sections. We're running through the last section. Should we carry on?
Oh, please.
Let's go.
I've got so much to do, and we've only got a little bit of time. As I said to you, I'm touching on questions, okay? I'm not answering all of them. I want to just show you the different types of questions, right, that you need to use. It's the skills that we're going to teach you. Okay, let's carry on. So we did biochemistry. All right, all the enzymes, the food tests, the carbohydrates, proteins, etc. We looked at the cell, right? When I finished with you, we were looking at the different phases of mitosis. So be able to identify from a picture which phase it is. Okay. Then the next section is mammalian tissue, and I find when it comes to the plant and the animal tissue, that is one of the sections that you battle with the most, because you need to be able to identify the tissue. If I give you, I'm going to show you, I've got a question here, all right, and you will notice it's different kinds of muscle, all right, tissue. If I go to the next question, all right, again, same part of it, different types of tissue. Can you identify from looking at the pictures the different characteristics that each of the mammalian tissues have, right? So if I were to go right to the first type of them, first of all, I give you that it's muscle, all right? I give you that it's muscle. So if I give you that it's muscle, you should now be able to know the 3 types. Skeletal, all right, or striated, cardiac, and smooth muscle. There they are. Now you need to be able to decide which of these is which. Okay. How do you decide? Quite easily. For some of you, I say quite easily being the teacher. Okay. If we have a look over here, striated means that we have dark and light bands. Now, this is always a problem when you come to these because both of those have got light and dark bands. Now remember, one of them is cardiac and one of them is striated. Which one is cardiac? Cardiac, you must always have bridges. Because why? Your heart needs to beat in a rhythm. All right? And if you're going to sing or dance whatever in a rhythm, all of the people involved must be in a rhythm. Do you see these little, lonesome little fibers? They're not reliant on a rhythm. Here, do you notice those fibers? Do they join? If they join, I've got a rhythm, okay? And that, because of that, I know that that is cardiac muscle. So this must be skeletal or striated. And these are single cells. They're also by themselves. No rhythm needed here. All right? And they are going to be your smooth muscle. So let's see type of questions I can ask. Which muscle tissue is voluntary? Voluntary means you control it. All right. Now if I have a look at the tissues, heart, do I control? No. All right. Brain does. Okay. Smooth muscle, are you sitting here going, breathe, digest, blood flow? All right, bladder, don't work. Right, probably doing that. Okay, but here you're going, okay, so voluntary, which one can you control? Right, now here, they haven't asked you, all right, which is which. So you can, if you're not too sure, you can answer the question by saying B. Right, but if you know, you can say skeletal muscle. All right, now they said, what does this mean? Voluntary means that you can control its movement. All right, so that's what voluntary means. Next question. Name 2 organs in the body in which muscle tissue is found. Now, number C. Now, that also gives you a hint because they're telling you 2 places. I don't have 2 hearts, all right? So they're telling you already that is not cardiac muscle. I want 2 places where I'm going to find it. Where am I going to find it? I'm going to find it my whole digestive system because it's smooth muscle. From when I start to swallow, okay, goes down, stomach, squish, intestines, all right, squish. Where else am I going to find it? Breathing, my diaphragm. I never know how to spell diaphragm. I apologize if it's wrong. Right, what else? All my tubes of my secretories. Right, my kidneys, my ureter, my urethra. All right, the tubes that the urine goes down. Going to squeeze all the way along. Okay, so we find smooth muscle. That was your hint. What 2 organs? Can't find in the heart. And you can't. It's in your muscle. Your skeletal muscle is muscle. Muscle is the organ, all right? You haven't got all these multiple different kinds of organs. So the question alone, all right, should have been given to you. Okay, I'm going to point up there. You saw where I labeled. Identify muscle tissue A and state how it's adapted for its function. Identify it first, all right? Then how is it adapted for it? It is... right, cardiac, and as I said to you, what's the most important thing, right, is these bridges. Okay, now we need to get a bit biological. I said to you they joined, right, for the rhythm. What are those called? All right, they are called intercalated discs. Intercalated discs or bridges, all right. And those bridges join. And when they join, all right, the heart gets its rhythm. Okay, the last one, right, is label structured X. Let's go and see what X is. X over here, can you see? X is the light portion and Y is the dark portion. And when we look at a muscle, right, what is number X, as well as one of the protein fibers that would result in the striated appearance? X is myosin, okay? That's the protein, myosin and actin. All right, and identify the structure labeled X. It's that whole thing. It's your myofibril. But do not worry too much about the structure of a muscle. You're going to do that next. You're going to do it when you do the skeletal system. You're also going to do it a bit next year. Okay, again, have a look. All right, have a look at the questions. From here, you should notice that is bone concentric. Over here, these are got little cilia, all right, and you can see it's ciliated epithelium. They lie next to each other. This, the cells, all right, is blood. So those are going to be your white blood cells. Okay, so the different kind of questions that you can ask there. I'd be able to identify, where do I find them? What is their function? Oh, I need to sneeze. I need to squeeze the nose here. Okay. Plant tissue, the same. Because you did plant tissue and you do plant organs, I can combine the two sections together. Okay. This looks a bit strange, but let's read what it says. It tells me the diagram below represents a portion of a young stem. Right, we looked at plant organs. You need to know dicotyledonous stem. All right, we looked at the structure of the root. We looked at the structure of the leaf. Leaves, roots and stems are all made up of plant tissue. So when I look at this diagram, all right, when I look at this diagram, not only am I asking you, all right, to know the plant organ, I can ask you plant tissue questions as well. Okay, so where they ask you, the first question is, list the numbers of the parts which strengthen the stem. What do we need to know? We need to know plant tissue. What plant tissue strengthens? All right, and what am I looking for? What's my strengthening? I'm looking for xylem. I'm looking for parenchyma. I'm looking for sclerenchyma, because all of those, all right, what is this? These cells are adapted. They thickened. And because they thickened, they are strengthening function, all right? So list the numbers. If we were to label, this is the epidermis, which is made of epidermal cells. Underneath it is parenchyma. Is it one of my strengthening tissues? Yes, so number 2 is a strengthening tissue. This is my vascular bundle. Remember the dicot stem, right? You did it round like that, secondary thickening, and you had a vascular bundle. And on top of that vascular bundle, can you see here, the cells are thick because they are made up of sclerenchyma. Is sclerenchyma a strengthening tissue? Yes, it is. Right? Now I'm looking for xylem. Where am I going to find xylem? Xylem. There we go. It's in, all right, in the vascular bundle. So what are my strengthening tissues? Right, and also, don't you think your mark allocation should have told you how many you were looking for? So number 2, number 3, and number 5. 2, 3, and 5. Number 2 was parenchyma, 3 sclerenchyma, and number 5 was my xylem. All tissues that are hard, all right? So here, again, as I said, it's a stem plant organ, but I want you to be able to tell me the plant tissues as well. Question number 2, identify the tissue mark 2 and make a labeled diagram to show one whole cell. Right, it's only 4 marks. They're not asking you to draw this whole biological cell. What is number 2? Number 2 is parenchyma, okay? Parenchyma. And why is it so nice to draw a cell? Because C, parenchyma starts with a C, and C stands for corners. And what on earth does that mean? It means parenchyma is strengthened at the corners. All right, so there's your cell. There's your cell. All right, you've got your nucleus there, and what do you notice? There's your thickenings. Don't have to draw this intensive biology. Draw one cell, right? And what is so special with parenchyma? Thickened at the corners. All right, last question here. Now, this question relates to the stem, not the tissue, but the tissue is going to come into play. It says to you, does the stem grow thicker each year? What is that called? Secondary thickening. All right, can you remember? All right, dicot plants, primary growth is upwards, and secondary, like me, all right, is when we grow outwards. Okay, and you need to have, all right, only dicots can undergo secondary thickening, and they must have their vascular bundles in a circle. Let's have a look at our diagram. Are my vascular bundles in a circle? Yes, they are. All right. So does the stem grow thicker each year? Yes. Give a reason for your answer. You can see the vascular bundles are in a circle, and also they are open. Right, my vascular bundles are open. What does that mean? The xylem has open. It can grow. Okay, there we're linking plant tissue growth, secondary thickening. All right, here again, here's a diagram of the leaf. What do we link with the leaf? Transpiration. All right, transpiration is movement. You have to do support and movement in plants. That is your plant organs. What is support in the plants? The parenchyma, the sclerenchyma. The support in animals we're going to do next paper. That's the skeleton. What's the transport in plants? Transport in plant is the water moving into the root, osmosis, up the stem, transpirational pull, capillarity, and root pressure, and water going out of, all right, the leaf. What is transport in humans? That is the circulatory system that we do in paper number 2. Okay, so again, your organs know the diagram of the leaf, know what moves in and what moves out of the leaf. Also, transpiration is a very, all right, can be asked as a data response. Again, you see graphs are quite popular when it comes to, all right, the life science people. If we have a look over here, let's just go through a few of the questions. It says, study the graph below, which shows the rate of transpiration. All right. Now, when you start with the rate of transpiration, that is the speed. How quick or how slow is it going to occur? All right. Now, when it comes to transpiration, what do we start? Things that start to need to go through our mind. We're looking at rate. Okay, what increases the rate? We're going to look at temperature. Be specific. Heat. The hotter it is, the faster transpiration will occur. What else? Wind. The more wind, the faster the increase the rate. All right. What else? Sunlight. The greater the sunlight, the greater the rate of transpiration. Okay. Then there's one that's going to decrease, and that is humidity. Obviously, the more water vapor in the air, the less we have. So as soon as you see rate, you need to think of the conditions outside. Is transpiration happening fast? Is it happening slow? All right. Now, when we have a look here at our graph, you will see it's a 24-hour clock, which means we have morning, afternoon, and night. And when we have morning, afternoon, and night, we have sunlight, and then we have no sunlight. Are we ready to?
Yeah, we're coming close to the
We're
end of our
30 seconds.
lesson.
We're coming. Can I just quickly?
Yeah, go for it.
All right, and then no sunlight. All right, and also what happens at night? It gets colder. All right, so have a look over here. Your transpiration will change. Is that you?
So guys,
Really?
looks like we've run out of time for today's lesson. I hope you guys got as much out of that lesson as you possibly could. Remember, all the notes we talked about today are on our Facebook page, facebook.com/learnextra. Thank you so much for the lesson.
Oh, it's a pleasure. Thank you.
I hope you guys got a lot out of it. So guys, remember, this show is proudly sponsored by MTN SA Foundation, and we'd like to give a big thank you to them, as well as this show being endorsed by the Department of Basic Education. And for any of those of you who are looking to get your learners, remember, you can go out and get the Safeway K53 guide, and this book is available in all major bookstores, as well as download the app off the Samsung App Store. We've got a big competition running with Chev, and you could win yourself a Chevrolet Spark L. So guys, until next time, keep well. I'll see you guys next time.